Imagine looking at a map of a bustling city.
Okay.
But, instead of streets and highways and subway lines, you're seeing the exact physical pathways of human motivation.
Oh, wow.
Yeah. You're seeing the specific neighborhoods where, like, a kind word from a friend lights up your day, or the hidden alleys where a substance can completely hijack your free will.
Right. And the really busy intersections where our deepest mental health struggles actually begin.
Exactly. Because, usually when we talk about a medical diagnosis, there's this expectation of absolute precision.
Yeah, like engineering.
Right. Like you break your arm, the x-ray shows that jagged white line, and the doctor just points to the screen and says, well, there it is. That's the problem.
It's comforting because it's binary. I mean, the bone is broken or it's not broken. We just naturally gravitate toward things that we can neatly categorize, things we can actually see.
But then you step into the world of human behavior or mood disorders or addiction, and suddenly that x-ray machine feels completely useless.
Totally useless. Yeah.
For the longest time, we've been looking at a diagnostic landscape that is, honestly, it's incredibly murky. Like, we talk about chemical imbalances or reward pathways, but it often feels like we're just gesturing vaguely at the whole brain.
It really has been the absolute definition of diagnostic muddy waters. But today, the water gets a whole lot clearer. We are looking at a fundamental shift in how we actually see the brain's geography.
Yes, we are. Welcome to the Deep Dive. Today, we have a truly monumental source to guide us. It's a 2026 paper published in the journal Neuron.
By Ravichandran, Bach, and a pretty massive team of researchers.
Yeah, and the title is quite a mouthful. It's spatiomolecular mapping reveals anatomical organization of heterogeneous cell types in the human nucleus accumbens.
Which is exactly why we are going to break it down.
Right.
Because behind that very dense academic title is essentially the holy grail of neuroscience.
So our mission for this deep dive is simple in concept, but huge in scale. We're going to explore the most detailed, high-resolution map ever created of the human nucleus accumbens.
Your brain's reward center.
Exactly. We're going to look at how this tiny structure regulates everything from, like, learning to avoid a hot stove to the crushing reality of severe opioid dependence. Okay, let's unpack this.
I think the best place to start is the territory itself.
Okay.
The nucleus accumbens, which we'll often just call the NARHAC, sits deep in the center of the brain. You can think of it as the core engine of our mesolimbic reward pathway.
The engine of reward. Got it.
Right. And its main residents are a specific type of cell called medium spiny neurons, or MSNs.
And just to paint a picture for you listening, they're called spiny because if you look at them under a microscope, their branches are covered in thousands of tiny thorn-like protrusions.
Yeah, exactly.
Those thorns act like little satellite dishes catching incoming signals from all over the brain.
That's a great way to visualize it. Now, these medium spiny neurons are GABAergic.
Meaning they use GABA.
Right, the neurotransmitter GABA, which means their primary job is actually inhibitory. They secrete GABA to dial down activity in other parts of the brain. They act as the brain's brakes.
Right. And classically, neuroscience has divided these MSNs into two very simple camps, like the textbook version.
Right, the classic D1 and D2 pathways.
The go and the stop.
Yes. So the DRD1 neurons, they express the D1 dopamine receptor. That is your direct pathway, generally considered the go signal for behavior.
Okay.
And then you have the DRD2 neurons expressing the D2 receptor. That's the indirect pathway, or the stop signal.
It's so binary.
It is. For decades, every textbook, every lecture on addiction or motivation has operated on this basic binary. Go or stop. Press the gas or pump the brakes.
But this paper, it entirely shatters that binary. It tells us that looking at the reward center as just a simple go-stop machine is like, I don't know, looking at a modern smartphone and calling it a telegraph.
It's way more complex.
Way more.
Yeah.
And the researchers figured this out using a combination of two incredible technologies on 10 post-mortem human donor brains.
The methodology here is really what makes this a landmark paper. To build this new map, they had to solve a massive problem. Historically, if you wanted to know the genetics of brain tissue, you had to use single-nucleus RNA sequencing, or snRNA-seq.
Right, which sounds complicated, but basically.
Basically, this involves taking the tissue, isolating over 100,000 individual cell nuclei.
103,000 to be exact for this study.
Right, exactly. And then reading their exact genetic mail, meaning looking at what RNA messages were actively being printed at that moment.
Like checking their outbox.
Yeah. This tells you exactly what type of cell you're looking at. And doing this, they found 20 distinct cell types in this tiny region alone, 13 neuronal and 7 non-neuronal.
But there's a major flaw with that sequencing technique, right? You literally have to blend up the tissue to read the mail.
That's the problem, yeah.
So what does this all mean? It's like the snRNA-seq gives us the exact hyper-detailed list of ingredients for a recipe. The flour, the sugar, the eggs.
Right, you have the list.
But because you blended it up, you have no idea what you're actually baking. You lose the architecture. Is it a layered cake? A batch of cookies? You just don't know.
And that is exactly where the second technology comes in. It's called spatial transcriptomics, specifically a tool called Visium.
Okay, what does Visium do?
This technique allows researchers to take a microscopically thin slice of the brain, lay it on a specialized grid, and read the RNA messages right there in place.
Oh, in place. So it shows you the final baked cake.
Exactly.
It reveals exactly where the frosting goes, where the filling sits, and how the layers stack up in three-dimensional physical space.
And what's fascinating here is what happened when they combined that ingredient list with the 3D map of the baked cake. They realized the old textbook binary of one go cell and one stop cell was completely wrong.
Completely.
They found 4 highly distinct subpopulations of the D1 go cells, which they just named A through D, and they found 2 distinct subpopulations of the D2 stop cells, A and B.
And these aren't just minor variations, right?
No, not at all. For instance, the D1 subtype A is actively using a gene called CALB1.
CALB1, okay.
While D1 subtype C is running a completely different genetic program based on a gene called RXFP2, they are structurally and functionally different machines living in totally different neighborhoods.
Okay, before we dive into those neighborhoods, let's just pause and do a quick 5-minute recap to make sure we have our foundation solid, because I know we just threw a lot of biology at you all.
Good idea.
So if I'm tracking this correctly, bullet point 1, we are looking at the Nucleus Accumbens, the brain's reward center. Bullet point 2. For years, we thought it was just a simple system of D1 go cells and D2 stop cells.
Right.
And bullet point 3. By combining essentially a genetic ingredient list, the snRNA-seq, with a 3D architectural map, the Visium tool, scientists discovered a massive hidden complexity. 20 different cell types operating in 3D space.
That is the perfect foundation, because now that we have this incredibly detailed map, we have to talk about how these cells are actually arranged.
Wow.
Historically, anatomists would look at the Nucleus Accumbens under a microscope and divide it into 2 strict physical regions, the core and the shell.
Sounds a bit like an apple. The core in the center, the shell wrapped around it.
Exactly. The field operated on that apple analogy for a long time. The assumption was that the core handled one specific job, maybe initiating physical motor actions.
Like grabbing a glass of water.
Right, and the shell handled a completely different job, like processing emotion or the euphoric reward of a drug. But when this research team laid their new high-resolution genetic map over the physical tissue, those rigid borders just vanish.
They found what they called spatial domains, or SPDs. By analyzing the tissue mathematically, they did find an area, let's call it the MSN1 domain.
Right, which sits on the lateral or outer edge. It kind of roughly resembles the old core.
And it expresses genes like CLB1 and ADOR2A.
Exactly.
And then they found another area, the MSN3 domain, sitting on the medial, or inner side, resembling the old shell, expressing a gene called CART-PT.
But the crucial detail here is that there was no wall between them.
No wall at all. To prove this, they used an algorithm called meringue.
Yeah, meringue, which is a fun name.
It is. And the meringue data prove that gene expression across this reward center doesn't operate in rigid boxes. It operates like weather patterns.
I love that visual, like looking at a meteorological heat map on the evening news, watching a storm front slowly roll in.
Right, exactly. That's the reality of the brain. They found continuous transcriptional gradients. The genetic programming of the cells flows smoothly from one side of the tissue to the other.
Yeah.
One specific pattern of genes, like the MCP3 gradient, would start incredibly strong on the intermedial side, and then slowly, gradually fade out as it moved toward the outer lateral edge.
And another pattern, like MCP4, would do the exact opposite, washing over the tissue from the outside in.
It's like the cells are essentially bathing in a continuous flowing soup of genetic instructions.
They really are.
But here's where it gets really interesting, and I have to push back a little bit on this.
Oh, hey, go for it.
If you're telling me it's all just a continuous flowing gradient, like a blurry genetic soup with no hard borders, how does the brain actually get anything done?
What do you mean?
I mean, if I need to jerk my hand back from a hot stove, or if I feel a sudden craving for sugar, I need a specific, sharp action to happen. Isn't a gradient too messy for that?
It seems like it would be,
It-
but if we connect this to the bigger picture of how human motivation actually works, you'll see why a gradient is essential.
Okay.
Think about how you tune an analog radio. You don't just want a digital switch that abruptly jumps from 90.1 FM directly to 100.5 FM. You need the dial to slide continuously so you can catch the exact frequency, the perfect nuance of the signal.
Oh, I see.
Human behavior is rarely just eat or don't eat. It's often, I'm slightly hungry, but I can wait, or I'm stressed, so I'm craving something specific. Gradients allow for that highly complex analog integration of signals.
So it gives our reward system a dimmer switch instead of a clunky on-off switch.
Exactly. It's a dimmer switch.
That makes total sense. We need nuance to survive. And what really blew my mind reading this source is that it's not just the neurons participating in this analog tuning.
Right.
The glial cells are deeply involved.
Yeah, for a long time, glial cells, things like astrocytes and oligodendrocytes, they were dismissed as just the brain's packing peanuts, just structural glue holding the important neurons in place.
But the researchers used a mathematical tool to basically deconvolute the tissue, sort of like taking a sip of a smoothie and being able to calculate the exact precise ratio of strawberry to banana in that one single drop.
And by doing that, they proved that these glial cells are not randomly scattered packing peanuts at all. They specifically cluster in certain areas, actively cuddling up next to very specific neuron types.
Like which ones?
For example, they saw glial cells aggressively clustering alongside the D2 subtypes A cells on the lateral side. This means the structural support environment of the brain is actively modulating specific synapses in real time. It's a fully integrated microenvironment.
Mind-blowing. Okay, let's pause for our second 5-minute recap to digest this layer.
Sounds good.
Bullet point 1. The old idea of the brain's reward center being neatly divided into an apple-like core and shell is dead.
Dead and gone.
Bullet point 2. Instead, using the meringue algorithm, we see these beautiful, continuous gradients of gene expression flowing like weather patterns, which gives our brain the analog dimmer switch it needs for complex motivation.
Yep.
And bullet point 3. The brain's support staff, the glial cells, are actually active managers, grouping up laterally with specific D2 neurons to fine-tune our behavior.
You've got it. But as with everything in neuroscience, there is a magnificent contradiction here.
Oh, of course there is.
Because despite these flowing, blurry gradients we just talked about, the map also revealed something jarringly specific. Hidden within the gradients were anomalies. Highly concentrated, distinct structures that look almost like archipelagos on a map.
Treasure islands.
Biologically speaking, yes. The researchers named them the D1 interface islands.
Let's talk about these islands, because this feels like a massive breakthrough. They are located along the bottom middle edge, or the ventromedial border, of the nucleus accumbens.
Right.
And the detail that stopped me in my tracks is that these islands are evolutionarily conserved. You can look at the brain of a rat, you can look at the brain of a macaque monkey, and you can look at a human brain, and you will find these exact same cellular islands.
And that's huge, because when nature conserves a physical structure that meticulously across millions of years of evolution, it is telling us something very clearly. This structure is absolutely vital for survival.
So what are they made of?
These islands are made exclusively of two highly specific subtypes of those D1 Go cells we mentioned earlier. Specifically, the D1 subtype B, which expresses a gene called RXFP1, and subtype D expressing CPNE4.
Okay, but what are they actually doing sitting there on the border?
To figure this out, the paper dives into how cells text message each other. In biology, they call it ligand-receptor interactions.
Ligand-receptor. But really, it's just a system of keys and locks.
Oh.
Right.
Exactly. The ligand is the physical chemical key floating through the brain fluid, and the receptor on the outside of a cell is the lock it fits into.
And they mapped this using an algorithm called Liana, right?
Yes. The researchers used Liana to essentially wiretap the tissue to see where the keys were being minted and where the locks were installed. And they found that these D1 islands are densely packed with a very specific lock called OPRM1.
OPRM1, the mu-opioid receptor.
Ah, that's the one.
For anyone listening, this is the exact biological lock that responds to our body's natural endorphins, like a runner's high. But it's also the exact lock hijacked by external drugs like morphine, heroin, and fentanyl.
Yes. These D1 islands are essentially hyperdense hubs of opioid reception, and they just sit there in the tissue acting as specialized receivers.
Waiting for keys.
Exactly. The surrounding cells, the different neighborhoods we talked about earlier, they're constantly manufacturing chemical keys. One key is a neuropeptide called PTIC,
Okay.
Yeah, sent out by the local D2 stop cells, subtype A specifically. Another key is PDYN, sent out by nearby D1 subtype A cells.
So if I can try an analogy here to help visualize this, it's like these D1 islands are incredibly powerful, highly localized cell towers standing in the middle of the city.
I like that.
They are absolutely covered in these OPRM1 opioid receivers, and their entire job is to catch highly specific text messages, the PNK and PDYN keys, from the surrounding cellular neighborhoods.
Exactly.
And depending on what messages they catch, they regulate our most intense feelings of pleasure, pain relief, and hedonic reward.
It's a brilliant system. But this raises an incredibly important question. What happens when the physical infrastructure of that specific cellular neighborhood is genetically compromised?
This part of the paper gave me chills. The researchers took massive data sets of known genetic risk factors for human diseases, basically a heat map of genes that we know contribute to mental illness.
Right.
And they overlaid that disease map onto this brand new 3D architectural map of the reward center. They essentially asked, where do the genes that cause psychiatric disorders actually live?
And the results were profound. They found that the genetic risk for schizophrenia, bipolar disorder, and severe depression doesn't just wash over the whole brain evenly. It is heavily, significantly enriched right inside those precise D1 opioid islands.
Wow. While other traits lived in totally different neighborhoods, like the genetic risk for neuroticism mapped strongly to that outer lateral domain, the MSN1 area we talked about, and the genetic risk for heavy alcohol intake mapped to the inner medial domain, MSN3. We're literally looking at the physical geography of mental health.
We really are. Let's do our third quick recap here because the implications of this section are massive.
Take it away.
Bullet point 1. Amidst the flowing gradients of the reward center, there are these ancient, highly specific D1 interface islands that are absolutely packed with OPRM1 opioid receptors.
Bullet point 2. They act like cellular cell towers. Using the Liana algorithm we saw, they catch PDYN and PDYN chemical messages from surrounding cells to regulate pleasure and reward.
And bullet point 3. Crucially, these exact islands are the physical ground zero for the genetic vulnerability to severe psychiatric conditions like schizophrenia, bipolar, and depression.
Okay, so we have our map. We know where the neighborhoods are, we know where the specialized cell towers are located, and we see how mental health vulnerabilities map to them.
Right.
But up to this point, we've only been talking about neurotypical, sober human brains. How do we know what happens to this beautiful, delicate architecture when highly addictive, powerful drugs enter the system?
Well, that is the ultimate test of the map. Now, obviously, we can't ethically induce severe life-altering drug addiction in human subjects just to study their brain tissue.
Right, that would be highly unethical.
So the researchers used a computational technique called transfer learning. They started by taking extensive single-nucleus RNA data from rats.
Right. They took rats, exposed them to drugs like morphine and cocaine, and then mathematically analyzed the rat brains to see what genetic programs the drugs turned on.
Using a tool called non-negative matrix factorization, or NMF.
NMF, right. So imagine listening to a massive, chaotic symphony orchestra. The researchers used this NMF mathematical algorithm that can listen to that wall of sound and isolate just the sheet music for the cellos or just the sheet music for the oboes. It extracts the hidden underlying programs.
That's a great way to explain it. In this case, the symphony is the chaos of a brain on drugs. The algorithm isolated the specific genetic sheet music that the drugs triggered.
What do they find?
Well, for example, when a rat was given an acute sudden dose of morphine, the algorithm found a program called NMF12.
NMF12.
Right. And this drug didn't just cause a temporary high. This NMF12 program spanned all the MSN domains and actually promoted neuron survival and altered dopamine signaling using a pathway called BDNFTRKB.
Wait, so it's helping neurons survive?
Yeah, it essentially fertilized the brain to aggressively wire in a new habit. And at the same time, another program called NMF3 triggered a massive cellular stress and metabolic panic response.
So that's for acute, sudden morphine. And what about chronic use? When the rats were allowed to self-administer morphine over time, simulating volitional addiction.
For chronic use, a completely new genetic program appeared called NMF23. This one literally alters thalamostriatal patterning.
Okay, to put that in plain English for our listeners, chronic opioid use physically changes how the brain routes signals. It's not just a software update. It rewires the hardware circuit board.
Exactly.
And what about cocaine?
For cocaine, the drug drastically hijacked the dopamine and glutamate systems. It upregulated genes like PDE7B and GRN1 in those specific outer and inner domains, MSN1 and MSN3, of the reward center.
Okay, I have to play devil's advocate here for a second.
Please do.
They took a rat high on cocaine or morphine, mathematically extracted its active genes, and essentially copy-pasted that onto a sober neurotypical human brain. How can we possibly trust that a rodent's brain maps cleanly onto complex human architecture? Aren't we making a massive leap?
It's a completely valid challenge, but it actually highlights the brilliance of establishing this baseline human map first. We aren't saying the human brain is the rat brain.
Okay.
By projecting the rat data, the algorithm is asking a very specific question. Where in the human brain does the exact physical machinery exist to play this specific song? And it found perfect matches.
Oh, I see.
It showed exactly where the human brain possesses the latent architectural vulnerability for these specific drug responses.
So it proves that our baseline circuits, our normal everyday neighborhoods, are physically pre-wired with the exact sockets that drugs like cocaine and morphine plug into. The vulnerability isn't just behavioral, it's deeply structurally physical.
Exactly. The drug doesn't invent a new pathway out of thin air. It ruthlessly hijacks an existing, highly specific spatial domain. For example, the cocaine signature didn't just light up the whole human reward center randomly.
Where'd it go?
It specifically found its home in the outer lateral domain, and it aligned perfectly with human cells that express a specific gene called TC1.
TAC1. And the reason that is so important is because TAC1 is a gene heavily linked to stress-induced anhedonia.
Yes.
For those of you listening, anhedonia is that terrifying inability to feel pleasure. It's the gray, soul-crushing flatness people experience during drug withdrawal, where literally nothing in life brings joy anymore. And this map shows exactly where that suffering originates on a cellular level.
It is breathtaking science. It fundamentally changes how we understand the architecture of reward, motivation, and disease. It proves that it's not just what genes are active in the brain, it is exactly where they are active in 3D space that dictates our human experience.
It's incredible. Over the course of this deep dive, we've gone from looking at this tiny part of the brain as a simple binary ghost-op machine to seeing this hyper-complex, beautifully intricate 3D landscape.
A landscape made of flowing analog gradients.
Right, surrounded by supportive local glial cells, and punctuated by these ancient opioid-rich interface islands that hold the keys to our deepest mental health vulnerabilities.
Yeah, we finally have a map that respects the true complexity of human biology.
Which brings us to the end of our journey today. But we want to leave you, listening, with a final provocative thought to mull over. We've talked about all these specific locations, right?
Yeah.
So if our brain's reward center has such hyper-specific spatial locations, like those D1 islands acting as dedicated, localized opioid cell towers, what does the future of medicine look like?
That's a great question.
If you have a genetic vulnerability to depression or addiction living in one tiny specific archipelago of cells, could the future of treatment involve targeting a physical location in the brain with a highly specialized intervention, rather than just taking a pill that floods your entire brain with medication and just, hoping for the best?
I think we are standing on the edge of moving from systemic flooding to absolute precision targeting. It's going to change everything.
Thank you so much for joining us on this deep dive into the map of human motivation. Keep exploring the landscapes of your own minds, question everything, and remember, sometimes the murky waters finally clear up, revealing a map of a city you never knew existed. Catch you next time.